I'm just going to do a brief introduction to the seminar series for anyone who's not familiar with it. This is the Tools and Technology Seminar Series. It's basically a venue to discuss tools, technologies, methodologies. They might be recently developed, they might be currently in development, they may not be developed by the speaker at all, just something that they're using and are of interest. I am scheduling speakers for next semester, so if anyone is interested, please feel free to contact me. We do have some open slots still. So this is we've always done this seminar series in a hybrid sort of atmosphere like this, but we're sort of flipping it a little bit, and that we may actually end up with more people online than in the room. So we'll see how that goes this semester. In addition, some speakers like today's are online, but some will actually be in this room. So for those of you who are interested in attending in person and continue attending in person, you'll get to see some live speakers, hopefully. So today's speaker is Shengcheng from the Boyle Lab. And she's going to talk to us about RegulomeDB. And with that, I'll let her further introduce herself. Thanks.
Okay, thanks, Marcy. Can you guys hear me clearly? My voice good?
Yes.
Okay, cool. Yeah. Okay, so thanks for joining my talk. I'm Shengcheng from Boyle Lab. So I just graduated earlier this year, and I'm now doing a short postdoc in Boyle Lab. So today I'm going to start with some general overview on those regulatory SNPs and how to annotate them in the non-coding regions. And then I will introduce the RegulomeDB database developed from our lab. So this is a brief diagram showing the process of gene regulation. As many of you are familiar with, the transcription factors. So in this process, the transcription factors, in this case, are those activators, are recruited to bind with the regulatory elements in the non-coding regions, like those enhancer regions. And then the gene regulation will be triggered here to activate the gene expression level here downstream. So those regulatory elements in the non-coding regions can also be those silencers. So in that case, the repressors, as another type of transcription factor, or TFs in short, will be bind with those silencers regions and then downregulates the gene expression level downstream. So when a generic variation occurs within those regulatory elements, so like the case shown here, this is a type of SNP, single nucleotide variants, as the most common variation for genetic variants. So this case showing here is the situation when the allele changes from A allele to a C allele here. And this can actually change in the binding status for the binding with the TF here. So like this case, if it's an A allele, it will have a perfect binding with this TF here and triggers the downstream gene expression level here. But in other case, if it changes to C, it might be decrease the binding affinity with the TF binding site here. And so in that case, the downstream gene expression level may be decreased. There can also be other such as scenarios happen here, like it can, in other case, increase the binding affinity with the TF here and will increase the gene expression level. Or it can even create a novel binding site with another TF. And this will have effect with the gene expression level from other regions instead of the gene here. So why we care about those regulatory elements in non-coding regions? Because actually many of them are associated with diseases. So like the results from GWAS studies, which essentially is comparing the allele frequency from the patient's group versus the non-patient's group for different disease or different traits. So like the case shown here, this particular SNP is found associated with the disease. So in the non-patient's group, you can see the T allele frequency is about a half across the population in non-patients, but in the patient's group, the T allele frequency is actually much lower than the non-patient's group. So this implies this SNP is associated with a tested disease, and so it is kind of SNP you will find from those GWAS studies associated with your studied disease. And actually more than 90% of the variants found from GWAS studies are in the non-coding regions. So some of the variants are validated to be regulatory variants that cause the disease, but many of those non-coding variants from GWAS actually remain underexplored. So one reason is because the GWAS study is essentially a type of association study. So it is actually hard to distinguish which SNP is actually the SNP that caused the disease. So like the case here showing there's a cluster of SNPs, they are in high LD, which means they're integrated together across the population. So very likely they will be having the similar level of association with your study disease. But it is hard to distinguish which one is really the variants that cause the disease. So the challenge here is really to identify the disease causal regulatory variants in the non-coding regions from associated studies like the GWAS studies, because like in many times the association doesn't necessarily mean the causal relationship here. So this is just showing you a more particular case of the study from associated studies. So like in this case, some studies found the SNPs in this particular locus on chromosome 1 are associated with changes in cholesterol levels. So in the studies, actually, this particular SNP is used as an index SNP for the association study, which is like a probe basically used in those kind of stats studies. But the following studies actually found this SNP, which is apart from the index SNP from like 1 kilobase apart. This is actually the regulatory variants that affects the gene expression level in liver tissue by creating a TF binding here in this site for the variant. So the idea here is that from those association study, very likely you will come up with a candidate list, usually consists of like hundreds of candidate variants, but it is hard to distinguish which one is really caused the disease. So is there any like experimental evidence or other source of evidence we can look at to further narrow down the candidate list from those association studies? So some things we can look at here is first to look at the sequence context. So we know that different TFs have different binding affinity across the DNA elements. It is usually represented by this position weight matrix, or PWM in short. So like the case here showing a particular PWM for TF binding region, like the fourth position here will have the most strong binding affinity for A allele, and the case is different for other positions within the binding motif. But another challenging thing for identifying regulatory variants is that there also depends on the epigenetics factors around them. So like here, the first we can look at is the chromatin accessibility. So we know that across the whole genome, some genome regions have like closed chromatin status, while the other regions are more accessible for TF bind with the regulatory elements. So like the here, basically showing this region as an open chromatin regions. And also there's histone modifications. So different genomic regions have marked with different histone marks. So we know that H3K4 trimethylation is usually a histone mark associated with enhancer regions, while like H3K9 trimethylation or other histone marks are associated with more like closed genomic regions here. And also there's chromosome looping. So because the enhancer regions and the target genes can sometimes be hundreds of kilobases apart. So the regulation between those two elements will be dependent on those chromosome looping, which can bring them to get closer together in the spatial distance here. So this is usually rely on the protein like CTCF or other cohesion proteins. So basically the sequence context plus those epigenetic factors contributes to the dynamics of gene regulatory network across various tissues or environmental conditions. So there are many functional genomics assays can help us to characterizing those regulatory elements based on those different epigenomic factors. So for open chromatin regions, as you may heard of, there's like DNA-seq can basically capture those open chromatin regions across in a genome-wide scale. And for histone modifications or TF binding sites, there's ChIP-seq assays can target either the different histone marks or different TFs binding sites. So the DNA footprint is basically a computational method to further identify those dips from the DNA-seq assays. So basically, it provides a more basically identifies those TF binding sites with a more high resolution than the DNA-seq assays. And then there's also assays looking at the gene expression levels variations. So the EQTLs data basically also can provide us some additional evidence on identifying those regulatory variants. So there's like large projects providing a huge set of those available data in various tissues or cell types, like the ENCODE project and those roadmap epigenomics project. Also, there's a GTEx project providing those EQTLs data sets. So a common idea of developing computational methods to predicting regulatory variants using those different layers of functional genomics evidence is simply to overlapping the prior variant with those different layers of evidence. So basically, a prior variant, if it's overlapping like a DNA-seq peak, which means it's in an open chromatin regions, then it implies this variant is more likely to be a regulatory variant within the regulatory elements and changing the TF binding status. So basically by layering those different functional genomics evidence, and recently many tools are using machine learning methods to find a better way to incorporate those different layers of evidence. And also there are other tools using deep learning techniques to learn the regulatory grammars in various cells from different assays simultaneously. So basically, eventually, you can get a candidate list from those kind of tools, basically give you a prediction score list for your current variants. So the highest score basically means this variant will be more likely to be a regulatory variant that affect TF binding status and might cause the disease you're looking at from the association studies. So basically, this is a way to further narrow down your candidate list from the association studies. So the RegularDB database is the database developed from our lab. I'll first walk through this simple demo first, and then talk about how the scores or the data source in detail later. So this is the interface from our website. there's a query in box here. So you can type in your query variants in a list. It can be a list of DB SNP IDs from DB SNP database, or it can also be a list of coordinates for your query variants. And you can also query by a specific chromosomal region. So we will be outputting the scores for the SNPs within this region as the results list. So, so currently we have a maximum limit of like 100 to 200 input query SNPs at most from the website interface. So the case here is just showing you if we input this like 5 query variants here, then click the search bar here. It will initially showing you a list on your query variants, so the locations, the DB SNP IDs, and the rank score and the probability score. I will explain in detail later. But you can also download your query results as a BAT file or a TSV file with more details on how the evidence looks like. And then we also provide downloading option for all the pre-calculated scores for those common variants from DB SNP database. Essentially includes like 10 million variants here, because currently it's not so feasible to pre-calculate all the scores for the whole genome. So we currently provide this pre-calculated list on the common SNPs here. So if you click any of the fields here, you can see more details on your query variant individually. So like if I clicked one of the variant here, it will show you like how many peaks from those functional genomics assays it's overlapping with, and the rank score, the prop score, the allele frequency from different projects, and its nearby SNPs you're looking at. And if you scroll down a little bit further, you will see essentially 6 summary table here. Each can be clicked further to see more details, and I will show later. But it's essentially some summarized tables on which of the like ChIP-seq evidence is overlapping with your query variant or the other source of evidence you will be looking at for your query variant. So I want to first explain more in detail for how the rank score and the prop score here. So the rank score here is essentially a manually designed scoring schema here. So as I explained in the background, we are essentially looking at the evidence from DNA-seq assays and the TF binding sites from ChIP-seq data sets. So like the case here, this particular variant, you can see its position is overlapping with the DHS region, so open chromatin region from DNA-seq data. And it's also overlapping with the REST binding, so TF binding site, and also a DNase footprint from DNA-seq data. And it's also have a hit on the PWM matching here for the REST TF. So this gives this variant a 2A score here in this case. So this is the detail for all the different ranking score we have in RegulomeDB database. So essentially, the more evidence is overlapping with, the higher score it will get from this ranking score schema. So if you think for this ranking schema, it's essentially a manually designed decision tree. So looking at each different layers of evidence, and if it's matching with all those evidence, it will give a highest score here. So in this case, it's 1A. But we also want to further improve this ranking schema by using machine learning methods. So essentially, we incorporate all those binary features from the initial ranking schema. So like the 6 binary features here. And we also included some other numerical features to further improve the performance. So like the chip-seq signal, other than those binary features, they are numerical features, so better describe the profiles for chip-seq data across different assays. And also we include information content change in PWM and the DeepSeq score. So this is essentially our deep learning model that learn the different regulatory grammar for various cell types, and it has a functional significance score, basically a summarizing score to describe how distinguish how significant the changing for the query variant is. So the technique here we use is simply the random forest model. So essentially we are changing this manually designed decision tree into a bunch of like 500 decision trees here. So essentially each of the tree is using a subset of our total feature set, and it's trained on a subset of our trained data set. So this is basically a technique to avoid overfitting and also improve the model performance at the end. So the average vote from those 500 trees will be our final score. So the prop score I showed you before from our random forest training step. So yeah, so the score here is the prediction score from the random forest model. And here is a box plot where we compare our new score versus the original, the initial ranking score here. So essentially you can still see a trend here. The lower ranking score initially, it also has a lower new prop score here. But the idea is now we can have like better distinguish for the variance in the same score category initially. And we have also tested the new score on some NPRA, so essentially validated variants from experiments, and show they have improved the prediction accuracy with the new prop score. So RegulomeDB database essentially directly mirrors the ENCODE data portal. So for the human, for all the human data, they have we have over like 2,000 chip-seq data sets from different cell lines or tissues or other differentiated cells. And we also include over 1,000 DNA data from different biosamples or under different treatment or conditions. So all those genomic intervals are stored in an Amazon S3 bucket, and those bed files are indexed in Elastic Search to enable our efficient search against our query position from the query variant. So just go back to the summary table on the genomics evidence. If you click like each of them, like the DNAs table in this case, it will show you the overlapping peak from the DNA-seq and what's the dataset ID and the derived organ, and some more details can be get if you click on the link here. So for the chip-seq data, we will show you like the binding TF for each experiment. So a summary plot here, and also more details can be get if you clicked them. So there's also a genome browser view for you can move your window here, so you can see the nearby genes overlapping your query variant, and also how's the you can click or filter those different peaks from different by the assays or by the cell lines. So cover closer view to see how the signals looks like from different assays. So I also want to talk about some of the work we are currently working on for further extending RegulomeDB prediction score. So one aspect, we are extending those scores to further predict regulatory variants in a tissue or organ-specific context. So the idea is here, so currently when query over those functional genomics annotations, we are just simply grabs all evidence across different tissues or different cell types. So this is essentially a generic score we will get from the random forest model. But we also, here we also further to query it in a tissue specific way. So we further look at what's the specific, in blood, particularly the query variant is overlapping, if it's overlapping with the DNA-seq peak or the histomark from enhancer regions or promoter regions here. So we designed another, we trained another random forest model here, and at the end have this tissue-specific score. So which is essentially a weight from the previous generic score. So predicts whether the variant is particularly functional in the tissue you're looking at. So like in the case is the blood tissue. So we also leverage those tissue-specific scores to an organ-specific context. So the idea is just simply combine all the assays or the annotations from the biosamples mapping to the same relevant organ here. So in this way, we are able to generate our organ-specific score across essentially 51 organs from ENCODE project. So all the purple labeled bars here are those organs we have available organ-specific scores from our algorithm. And this is basically a validated variant I showed at the beginning. So the one actually have effect on the cholesterol level changing in the liver tissue. And in this plot, actually you can, so on the x-axis is the different organs. So the 51 organs, we have the available organ-specific score. And this is basically the candidate list from the association study. And this validated variant, you can see actually it has the highest liver-specific score compared with the other candidate variants. So this is essentially a validation showing our TERF liver-specific score is able to pick out the actual regulatory variant across the candidate list. So we also validated the scores across the organs from more various organs from GWAS studies. So here, this plot is showing so each row is a GWAS trait from the GWAS studies we downloaded. And on the each column is the 51 organs we have the available organ-specific scores. So these are the Z scores shown here. So essentially, the trait has an enrichment of regulatory variants functional in the corresponding organ will have a highest Z score showing here. So like this is the top organ with the most enrichment for the regulatory variant from our algorithm. So just give you a closer look here. So we found actually that various traits are associated or most enrichment with their corresponding organs, like some coronary artery disease, they have the most highest enrichment in the arterial blood vessel organ, or like other the immune disease actually most enrichment in those immune relevant organs, and also some like blood pressure or cholesterol most enriched in liver tissue here. So another thing we want to further incorporate is the target genes. So we want to also tell user what might be the candidate target gene you are looking at for those putative regulatory variants. So we have come up with like 3 strategies here. So in a most naive ways, you can just look at the gene with the nearest TSS, so transcription starting site from your query variant. But we also want to incorporate with some topological evidence from test regions from like high-C data sets. So, so essentially, we can tell you like the genes, what are the genes in the same test with the query variants. So, which means they are close in the spatial distance. So also we want to incorporate the evidence from EQTLs. So from the query variant, which SNP is has the strong LD with the query variant, then its associated E gene will be the target gene for your query variant here. Okay, so I guess I end up with a little bit earlier, but essentially RegulomeDB data scores prioritizes putative regulatory variants in the non-coding regions. And we have this RegulomeDB web interface basically provides the interactive annotations on the non-coding variants. So showing you what's the specific evidence you are looking at that's overlapping your query variant. And we are also working on integrating our new organ-specific scores and target gene assignment into RegulomeDB. So hopefully can be implemented that in future. And we are also moving all our data sets from HG19 to HG38. So it's also in progress. So with that, I want to thank all the lab members from Boy Lab and also the collaborators from Cherry Lab, Ben, Yunhai, Emmer, and Pedro. They help us and work with us for designing the lab, the website interface. And I'm happy to take any questions. Thank you.
Thank you, Shengcheng, for that excellent talk. So if anyone has questions in the room, raise your hand and I can bring you the microphone. If you have questions online, then you can put them in the chat and we can read them from there, or you can use the Zoom controls to raise your hand and then unmute yourself. There's a question in the room.
Hi, this is Maureen.
Hi.
I was wondering how you define the gold standard to use when training the random forest model.
Yeah. So the golden standard data set essentially comes from the MPRA data sets. I don't have like a detailed slide here, but they are from massively parallel reporter assays. So basically we have data sets from 3 cell lines, GM12878, K562, and HEP-G2. So basically they are the standard, golden standard data sets we compare with, because there's like several previous studies also using this kind of data sets to compare with other tools, so we can easily to compare our performance with them. Yeah.
So it looks like there's a question in chat, and the question is, how often do you update RegulomeDB?
That's a great question. So we actually just updated for the 2.0.3 version. So the plan is actually to at least give it an update on a half of a year base. So yeah, so we are looking at like another updation soon after we incorporate those HG38 data sets. So hopefully within next year, I think so. Yeah.
Again, if anyone else online has a question, feel free to put it in the chat or to use the Zoom controls to raise your hand. So it looks like there's another question in the chat. So it says, there are lots of functional tools like yours. Can you list few unique features of yours?
Yeah, sure. I guess, yeah, there are like different functional tools. I think the first thing is basically the tools using machine learning methods. They are using like different types of training data sets. So like some of them are using basically more like disease relevant variants from like a DCMB database. So the others like from similar to us are using data sets from like in a more regulatory aspect. So like we are using those MPRA data sets, or we actually call some ChIP-seq validated, so allele specific binding SNPs from ChIP-seq data sets. So I guess that's one aspect. So I think our tools have more emphasis on specifically identifying those regulatory variants that are changing the binding status with TF binding sets in the relevant tissues. So this is one thing. And the other aspects I think is because we have a directory mirrored encode portal. So I think we have a large data set, and it can be updated in easily with the ENCODE project, as they are including more data sets for different tissues or different conditions. And also, so in the feature list I showed before, so part of the features that come from essentially overlapping those functional genomics assays from ENCODE project, but we also include DeepC scores, so the deep learning scores from the DeepC group. And actually, we found in our studies, so it gives benefit to actually combine the direct experiment evidence from ENCODE project, like the binary features, and also with those those features from deep learning techniques. So essentially, the deep learning, I think the deep learning techniques are providing the regulatory grammar from a large training set, but they may be they may have like low performance for those underrepresented cell lines from their training set. But our like binary features give like another complementary aspect to adding those experiment features directly apart from those deep learning scores. So.
Are there any other questions? Oh, it looks like another question came in via chat. So is your tool able to distinguish between lead SNPs, for which we have the most information from GWAS studies, and likely causal variants that would be segregating with the SNPs being measured?
Yeah. So I just want to make sure I understand your question. So essentially, the idea of our tool is to give a more like accurate prediction on those causal variants. So it's actually exactly the idea for our database. So so ideally, if you have a candidate list, which consists of the lead SNPs, and you can include other candidates SNPs like some SNPs in strong LD with the lead SNP, and input them all in our database. And the idea is our prop score will give you will prioritize those variants that are most likely to cause the disease, because we are looking at the other genomics evidence, like if it's particular binding with a TF binding site there, or if it's an open chromatin region. So gives you another layer of evidence besides the association evidence, which is like the lead SNP. So I don't know if that answers your question. Sure.
Sounds like that did answer the question. As, Dana wrote, excellent. Thank you for explaining the utility for lead versus associated SNPs.
Cool.
Just give it another moment in case anyone else wants to enter a question.